Good morning, everyone. In this video, I'll be talking about another different methodology used in genetic manipulation, that is genetic material transfer. In this case, simply what happens, the gene which will express or produce the product of our interest, of industrially important thing, okay, that will be transferred from one microbe to completely different microorganism. Okay, so this microorganism will be heterologous to this microorganism. Suppose you transfer a gene from bacteria to yeast. So obviously yeast and bacteria are heterologous organism. They are not similar, they are not same in any way. So gene transfer occur from one microorganism to completely different another type of microorganism. Okay, and that gene is allowed to be transcribed and translated in the new host microorganism. Okay, and that will expressed in the new microorganism, and this whole process is basically known as heterologous gene expression. Why heterologous? Because suppose you have a gene of interest that will give rise to production of antibiotic, and that gene will be transferred and cloned into yeast, okay, which will help to produce wine in the wine factory or wine industry. So yeast doesn't have any kind of antibiotic producing gene. Suppose the antibiotic producing gene is missing from the yeast in the parental strain, but the yeast that will give rise to good amount of wine, but there is a problem of contamination of the with the other microbes, so the yeast need a antibiotic. So how it will get that? Then this genetic material transfer technique will help the gene of interest. Suppose a bacteria that is producing a gene, antibiotic that will expressed by a gene, and that gene will be transferred and cloned into the yeast. So now yeast has both the microbial gene which will be transcribed and translated into the yeast, so there is no chances of contamination. One such example is Pediococcus pentosechias. Okay, Pediococcus pentosechias is a bacteria from which the gene that will produce Pediocin antibiotic. Okay, Pediococcus pentosechias, it's a bacteria which will produce this Pediocin antibiotic. Now Pediocin antibiotic expressing genes or gene that will be transferred into a yeast which can produce wine. Now these yeast has both the antibiotic producing gene transferred from this bacteria, and it also has the wine producing gene, which is the inherent property of the yeast, means it has the inborn property to produce wine. So both the genes are now present in this in this yeast. So now it become very powerful strain in the industry, wine industry. So this is basically the genetic material transfer. An advantage of this is, again, specific gene or specific genetic material only can be transferred by this method, which cannot be transferred by protoplast fusion method. I have already mentioned about it. So thank you so much. If you like, please subscribe my channel and share with your friends. Have a good day.